The nucleus is the hallmark of eukaryotic cell. A very term eukaryotic means having a true nucleus. Because in prokaryotic true nucleus is not present, there are only chromosome thread-like material in prokaryotes. The first part of nucleus is nuclear envelope. Nucleus is enveloped by a pair of membranes enclosing the lumen that continues with that of the endoplasmic reticulum. The inner membrane is stabilized by the meshwork of intermediate filament protein called lamin. The nucleus envelope is perforated by thousands of nuclear pore complexes, which are written as NPCs, that control the passage of the molecules and in and out of the nucleus. The next is chromatin. Chromatin is the nucleus content of the chromosomes of the cell. Each chromosome consists of a single molecule of DNA complex with an equal mass of proteins. Collectively, the DNA of the nucleus with its associated proteins, which is called as chromatin. Most of the proteins consist of multiple copies of all kinds of histones. These are basic proteins bristling with positively charged arginine and lysine residues. Both arginine and lysine have a free amino group on their R group, which attract proteins which attract protons, giving them positive charge. So just the choice of amino acid you would make to bind tightly to the negative charge phosphate group of DNA deoxyribonucleic acid. The chromatin also contains small amount of wide variety of non-histone proteins. Most of these are transcription factors such as steroid receptors, and their association with DNA is more transient. The image on the right shows that the five histones separated by electrophoresis. These five proteins may vary little from one cell to another or even from one species to another. However, there are many non-histone proteins in the chromatin do vary from one cell type to another and from one species to another. Two copies of each of the four kind of stones to H2A, H2B, H3, H4 form a core of proteins. The nucleosome core around this is wrapped about 147 base pair of DNA from 20 to 60 base pair of DNA link one nucleosome to next. Each linker region is occupied by a single molecule of histone 1. This region is longer adjacent to the promoter of genes, which presumably make more room for binding transcription factors. The binding of histones to DNA does not depend on particular nucleotide sequences in the DNA but does depend critically on the amino acid sequences of the histone. Histones are some most conserved molecules during a course of evolution. Histone H4 in the cough different from H4 in the P. plant at only two amino acid residues in the chain of 102. The above electron micrograph for nucleosomes show chromatin from the nucleus of a chicken red blood cell, but unlike most mammals, retain the nucleus in their mature red blood cells. The arrows point to the nucleosome. The formation of nucleosome help somewhat, but not nearly enough to make DNA sufficiently compact to fit in the nucleus. In order to fit 46 DNA molecules in human, totaling over 2 meter in length, into a nucleus that may be only 10 micrometer across, require more extensive folding and compaction interaction between the exposed tail of the core histone causes nucleosome to associate into compact fiber 30 nanometer in diameter. These fibers are then folded into the more complex structures whose precise configuration is uncertain and which probably changes with the level of activity of the genes in the region. The next is histone modification. Although their amino acid sequence primary structure is unvarying, individual histone molecules do vary in structure as the result of chemical modification that occur later into individual amino acids. These including adding acetyl group to lysine. Next is phosphate group to serine and threonine. Last is methyl group to lysine and arginine. Although 70 to 80% of the histone molecules are incorporated in the core, the remainder at the N-terminal dangles out from the core as a tail. Most of the chemical modification occur on these tail, especially of H3 and H4. Most of these changes are irreversible. Are reversible. For example, acyl group are added by enzyme called histone acyl transferases. HAT is not to be confused with the HAT medium used to make monoclonal antibodies and are also removed by histone deacetylases. Most often not, acetylation of histone occur in the region of chromatin that become active in gene transcription. This make a kind of intuitive sense as adding acyl group neutralizes the positive charges on lysis, thus reducing the strength of association between the highly negative DNA and highly positive histones. However, there is surely more to story. Acetylation of lysis 16 on H4 prevent interaction of their tails needed to form the compact 30 nanometer structure in inactive chromatin and thus is associated with active genes.